Yes, right. Hello and a very good afternoon to all those who are joined for today's HBNI webinar. We are very happy to welcome amidst us Dr. S.V. Chiplunkar, Subhadra Chiplunkar, who was formerly the director of ACTREC, part of TMC, which was part of HBNI, and she was also the convener of the Board of Studies in Life Sciences. She's had a very long innings in research in life sciences. She's guided a large number of students. It's a great privilege to have her with us. We know that she is also a very good teacher. So we keenly look forward to her lecture. Before I hand over the mic to her, I want to only inform you that HBNA is also planning 2 courses in the coming year. The announcements can be seen very soon in the Anuvidya website of HBNI. One on solvent extraction related topics by Professor J.B. Joshi, who is an emeritus professor of HBNI. He'll be talking about two-phase contactors and reactors, and it will be a course for a few months duration with about 40-50 lectures. We are also planning a course on nuclear science and technology, an introduction, being coordinated by Professor Tumor. This will also be very useful to researchers who plan to engage in programs related to radioactivity or radiation or nuclear science. So please look out for announcement in the website of HBNI. And as you are aware, the lectures given by our invitees as part of these webinars, they are all available on the YouTube under the channel, in the channel HBNI webinar. So you can view them subsequently as well. And we are glad that you have joined us with us today, and a very warm welcome to all of you. Now, without taking any more time of yours, I request Dr. Chiplunkar to deliver her lecture. Thank you. Thank you. Can you share that?
Thank you, Professor Rao, for this invitation. And I would like to thank the Honorable Vice Chancellor for the wonderful initiative of starting these webinars. I myself have attended some of them, and I've really enjoyed it. So today, friends, I'm going to talk to you about cancer and the immune system. I've called it the clash of the titans, because these are really two powerful forces acting against each other. Now, this is such a vast topic, and I'm trying my best to cover it in a limited time span. So you'd get a bird's eye view of things, but those who are deeply interested in the topic, they can further read them or send me emails, and I can send them interesting papers. So to begin with, let's understand what actually cancer is, and what do you mean by the term cancer and metastasis? Cancer is uncontrolled proliferation of cells when the tumors are proliferating and at a particular site, they then gain the ability to dislodge from the tumor site, from the primary tumor site, and lodge at other distant sites in the body. And this could be the lungs, the bone, or the liver. Now, this process is called as metastasis. The lodging of tumor cells from the primary site and getting and homing to other tumor sites, sites in the body. Now we know very little about what regulates the dormancy of these cells at other sites, the heterogeneity, and the tumor environment or the niche that is created for them to stay happily over there and form tumors later on. So this is a very complex process. And all this happening in the face of an active immune system or an immune response. I've just put across some warning signs for cancer. And these, you all know, these are change in bowel or bladder habits, a sore that does not heal, unusual bleeding, thickening or a lump, indigestion, difficulty in swallowing, obvious change in a wart or a mole, and nagging cough and hoarseness. These are signs, and if you notice these signs, please do take this seriously. Now, what are the cancer causes and risk factors? Cancer basically is a genetic disease. So there's mutation in the genes which are happening. There could be a family history of cancer. And the classical examples are breast cancer, ovarian cancer, colon cancer. You must have had, all of you must have heard about Angelina Jolie's case. So BRCA1 and 2 mutations. So family history plays an important role. Older age is another risk factor. Exposure to carcinogens in various forms, pesticides, or any other chemicals which a person gets exposed to. Chronic tobacco abuse or alcohol is another risk factor. Lifestyles, hormones, diet, environmental factors, exposure to radiations and various chemicals. Chronic inflammation. Chronic inflammation is when it's now known that cancers develop on a background of chronic inflammation. For example, chronic pancreatitis, which causes pancreatic cancer, which may lead to pancreatic cancer, or cholelithiasis, which can lead to gallbladder cancer, or Crohn's disease, which or ulcerative colitis, which may cause colon cancer. Immunosuppression is another risk factor. Patients who are on long-term immunosuppressants, like transplant donors, kidney transplant patients, or organ transplant patients, are at a risk of developing cancer. And of course, there are viruses which are known and associated with cancer. A classical example being the human papillomavirus, which causes cervical cancer, or the hepatitis B virus, which is associated with hepatocellular carcinomas. And of course, other infections like Helicobacter pylori, which is known and is associated with stomach cancer. Now, this is just, I want to share with you an interesting paper which came up in Lancet in August last month, a few months ago. And it talked about malignant cancer, which was diagnosed in dinosaur for the first time. The first evidence of a dinosaur living with a malignant cancer, which was discovered 77 million year old fossil. So cancer has been there with us for very, very long. And this opens up a really new area for research that cancers existed then. And those who are interested can read this paper, which was published in Lancet. Now, looking at the cancer incidence in India, and we refer to the Globocon, which is the IR data, it talks about the cancer incidence in Indian males and in females. And Indian males, the cancer which tops is the cancer of the oral cavity. And this is, of course, because of the tobacco abuse, followed by lung, stomach, colorectal cancers, and esophagus. While in the females, earlier it was the cervix, but now we have the breast cancer, which has taken over the cervix, followed by the ovarian cancer, and then other cancers. So these are the two dominant cancers, cancers of the oral cavity in male and breast cancer, cervix cancer dominating in females. Now, what is the difference between a cancer cell and a normal cell? There are a lot of differences, both ranging from shape, the nuclear cytoplasmic ratio, the way the cells communicate, blood supply, because tumors need a whole lot of blood supply, which is called as tumor angiogenesis, while blood supply is only needed in normal cells during the time of wound healing or repair. The glucose oxygen requirements are very different. Tumors love and crave for glucose, while glucose in the normal cells, glucose requirement is limited. The energy efficiency, and so on. There are several points which can distinguish cancer cell from a normal cell. What are the treatment modalities for cancer? As you all know, it's surgery, radiation, chemotherapy, and this doesn't follow the same chronology, because sometimes you have to debulk the tumor before surgery or give neoadjuvant chemotherapy to reduce the tumor size. So there are protocols, guidelines on how and when what would be used. What I want to point out is that the fourth arm is now immunotherapy. And this is emerging as a very dominant field for treating cancers. And today we are going to understand what this immunotherapy is and what are the challenges. We all know that 2018, the Nobel Prize in Physiology and Medicine was given to Tasuku Honjo and James Allison. They shared this for the discovery of cancer therapy by inhibition of negative immune regulation. So this was what we now call the checkpoint inhibitor antibodies, which block this negative regulation. And you'll come to know during the course of this talk what this actually means. But so cancer immunotherapy has now taken a center stage. It has created a renaissance in the treatment modality of cancer. And this is because it has brought about responses in treatment refractory cases. Patients who have failed all lines of treatment are treated with immunotherapy. So this is really a revolution or a renaissance which is in the field. And of course, I must say that there are only a subset of patients who respond to this. And so we need really good biomarkers to understand upfront which are the patients who would benefit from this form of therapy. Now, what are the different immunotherapeutic modalities? You have checkpoint inhibitor antibodies, and I'm going to explain to you soon what this checkpoint inhibitor is. You have monoclonal. These are basically monoclonal antibodies which are against these checkpoints, which are called as programmed death 1, PD1, and PDL1, the ligand. You have CAR T cells, the chimeric antigen receptor T cell therapies, and then, of course, the vaccines, the cancer vaccines. And these are already in the clinics. These are offered to patients and have shown remarkable responses. And these are either given as standalone therapies, but mostly now in combination with radiotherapy, combination with chemotherapy. The story of vaccines is still, I would say, that's a long way to go, but very interesting data coming from the preclinical data and from the phase 1, phase 2 trials. Now let's get back to the hallmarks of cancer. Hanahan and Weinberg in 2000 defined what are the hallmarks. And they defined that sustaining proliferative signaling or resisting cell death, inducing angiogenesis, enabling replicative immortality, activating invasion and metastasis, and evading growth suppressors. These are the cardinal signs or the hallmarks for cancer. They later on modified this theory, and in 2011 came about with two important modifications. They included avoiding immune destruction and tumor promoting inflammation. So these became very important hallmarks now, and which we will be talking about, and I'll be explaining to you about how the tumor evades being recognized by the immune system. And before we move on to that, because I understand that those who are listening to this talk may not be from the field of life sciences, or may not have a background of immunology at the underground or postgrad. And so I thought I would give you first a brief introduction to the immune armament that we have. So basically, our immune system is made up of two arms: the innate arm, which composes of the innate immune cells, and the adaptive arm. In the adaptive arm, you have the B cells and the T cells. These are basically lymphocytes. B cells make antibodies, and which can bind directly to the antigens. T cells are either the CD4 cells, which are called as its T helper cells, or the CD8 cells, which are the killer cells. And the T cells do not recognize antigens directly like the B cells, but they need another cell to present to them that antigen. And when I say an antigen, it could be a bacteria, a parasite, or for that matter, we're also talking of tumor antigens. So these have to be presented to the T cell by antigen-presenting cells. So please remember, T cells need processed and presented antigens to get activated and to mount an immune response. In the innate arm, you have several cell types. You have mast cells, dendritic cells, macrophages, natural killer cells, and granulocytes, which compose of basophils, eosinophils, neutrophils. And bridging these two, the innate and the adaptive, are two specific cell types called the gamma delta T cells, which are more like B cells in recognition of the antigens. They don't act like the classical T cells, though they are T cells. They can bind to antigens directly, and you have the natural killer T cells. So imagine the army the immune system has to fight against the cancer cell. Let's now look at a very interesting theory in immunology, which was put forth by Hans Schreiber and came out in 2002, which explains, and he called it the 3 E's of cancer immunoediting. It is called that the first phase is elimination. Tumors are erupting in you and me and everyone, but we have a very active immune system, which is composed of the innate and adaptive immune cells, which take care of these mutated tumor cells. And this theory is very similar to the theory of immune surveillance, which was put forth by Sir Macfarlane Burnet way back. So elimination is where the competent immune system is able to take care of these tumors. Now, as you know, the tumor cells have come from body's own cells. They're not like a foreign particle like a bacteria or virus. So naturally, and there is, of course, those who have studied this will understand it better, that there is thymic selection and self-reactive T cells get eliminated in the thymus. So these cells may not be highly immunogenic, and those which are weakly immunogenic, but not recognized by the immune cells, escape and go and enter. These weakly immunogenic tumor cells escape and go into the equilibrium phase, where they stay sort of dormant for a very long time. And this is a very dynamic process, and the immune system sort of shapes or sculpts this tumor. It allows the growth of these weakly antigenic tumor cells. So much so that a clone of these cells finally starts growing, and this escapes being recognized, or this is able to escape the immune attack and escapes. So this is this phase when the tumor becomes clinically evident. So this phase of equilibrium may go on for 10 to 15 years. This is a state of dormancy, where the tumor is not even evident. And only when the tumor goes to the stage of escape phase that it's able to grow unabated, evading the immune recognition. So there are two phases to the immune system. One, which is bright and good, which you are able to eliminate the tumor cells. And in the other phase, it is actually the immune cells acting against, not doing their normal role of protection, but in fact promoting tumor growth. And actually, when you see, and I'll show you in a couple of examples how these two are there present in the body, both immune surveillance and immune subversion. Now, when you talk of a tumor cells, these are generally infiltrated with lymphocytes. And a scientist called Jerome Gallon and Wolf Hermann Friedman, these are French, the French group, which coined the term immune contexture. They said that it is very important to understand where the lymphocytes are located in the tumor. And they said there are several things which are important. Which type of lymphocytes infiltrate the tumor? What is the location of the lymphocytes? Are they in the core of the tumor? Are they at the invading margin? What is the density of these lymphocytes? And what is their functional orientation? And are there tertiary lymphoid structures which are created in the tumor microenvironment? So there are several factors. And they said the final outcome will depend on how these cells are placed and how they behave in the tumor microenvironment. And they showed this with a very nice data, which was done on some 15,000 patients. They took several types of cancers, and they studied the CD8 cells, the tertiary lymphoid cells, the regulatory T cells, these are suppressor cells, the M1 and M2 macrophages, the M2 are the tumor-associated macrophages, and they looked at the responses in these patients. They showed, in short, that a positive prognosis is seen when you see lymphocytes infiltrating the tumors, various tumors, which have a CD8 T cell phenotype. And they also showed that these T cells have a memory marker. So if you have the tumor invaded by CD8 T cells with CD45RO marker, the prognosis is going to be very good. And the red dots, like the M2s or the Ms and the Tregs generally, it's not a favorable prognosis. So he coined the term immunocontexture to understand upfront what would be the response to therapy. It tells us a lot about how the tumor is going to behave. And based on this, tumors have been classified as inflamed tumors which have inflammation, which I spoke about earlier, and that these are hot tumors. And then we have cold tumors, which are non-inflamed. They do not have the right cytokine milieu. They did not have the expression, the right molecules on them. The hot tumors are the ones which respond well to immunotherapy, and the cold tumors are the ones which don't respond to immunotherapy. And the interesting question is, how can you convert the non-inflamed or the cold tumors to hot tumors so that they respond to immunotherapy? Now, again, I bring you back to the tumor microenvironment. In the microenvironment, you have the tumor cell, which is dominant, which is proliferating, secreting a lot of factors. And in fact, the tumor secretes factors to invite the suppressor cells, tumor suppressor cells, into the tumor milieu. So you have all sorts of suppressor cells, like the myeloid-derived suppressor cells, the T regulatory cells, and also the other cells which contribute to immune suppression, sitting in close proximity of other very active immune cells like the CD4, CD8 T cells, the gamma delta T cells, the DCs, the activated DCs, the NKs, the CTLs. So imagine a very, very complex environment and the crosstalk which is going on between the tumor cells and these cell types. So it's really a challenge for the immune cell to perform and to evade, and for the tumor cell to evade being recognized by the immune cells. So it's like a tug of war. And I like to show this slide because I feel these are really two big giants fighting. They are poised against each other, throwing challenges, and the outcome will depend on who is more smarter than the other. And unfortunately, most of the time, it is the tumor. So how has the tumor learned? What mechanisms does the tumor use to evade being recognized by the immune system? So the first one is inhibition of tumor antigen presentation. Or because a T cell, a classical T cell, will recognize an antigen. So tumor has the strategy of lowering the antigenicity or lowering the expression of MHC class 1, which is the most important antigen-presenting molecule on the tumor cell. The tumor has the ability to secrete immunosuppressive factors like transforming growth factor beta. And this transforming growth factor beta can dampen the activated immune cells. Or, as I said, the tumor has the ability to invite or recruit regulatory T cells, which are the immunosuppressive cells. So this happens through expression of several chemokines and cytokines, through which these Tregs get recruited from the peripheral blood into the tumor milieu. Or inhibition of immune attack by checkpoint pathways, and this is what I'm going to talk to you more in detail. Let's look at each one of them. We spoke about antigenicity. The tumor can downregulate the antigen presentation or even lower the antigenicity of the tumors. Tumors express a variety of non-mutated and mutated antigens that can elicit immune response. To avoid immune-mediated elimination, cancer cells lose their antigenicity. Because tumor is heterogeneous, it can, and mostly very low in immunogenicity. Immune selection of cancer cells that lack or mutate immunogenic tumor antigens, which I spoke to you in the 3 E's, that the immune system helps, in fact, to select cancer cells which are low in immunogenicity. And so it's a challenge to the immune system to go and attack these tumor cells. Because as I said, during thymic education, the strongly reactive self-antigen reactive T cells are eliminated. So only it is the T cells with low affinity which can interact with these antigens which are expressed on the tumor cells. And of course, there are defects in antigen presentation. In the next cartoon, I'll show you the defect at every level of antigen presentation in the T cell. And the tumor mutation burden predicts new antigen expression. This I've deliberately put here because this is an emerging area that mutations which occur, these are the non-synonymous mutations which are occurring in the tumor, which give rise to novel antigens, epitopes, which the T cells can recognize. But let's look at what happens, how the antigen presentation is defective. Now you know that when an antigen, it has to be processed and presented, as I mentioned to you, by an antigen-presenting cell. So what the tumor does, first, there is antigen loss, as the antigenicity of is reduced. Second, there is defect in MHC, the MHC molecule, which is a key molecule, loss of beta-2 microglobulin. all the machinery which is under LMP2 subunits of the proteasome, the transport associated proteins which are going to carry this MHC peptide complex to the surface. So all this is defective, and at every stage there is a defect. So carrying the to the membrane is hampered, and so the immune system is so able to look at or bind to the antigen which is presented on the tumor cell. And this has been elegantly shown through various immunohistochemistry which has been done on the tumors. Like this is an example of the colon tumor, which you see that the expression of the TAP protein is lost. Now, they've also looked at mutations in HLA class 1 genes in these colorectal cancer patients, and all the dark ones are the HLA loci. And if you look at it carefully, beta-2M, HLA, HLA-B, C, TAP1, TAP2, and NRC5 is a transcription factor associated, which is needed for the expression of MHC class 1. Everywhere you see mutations. So the tumor has mutated all these key molecules which are needed for antigen presentation. And this overall mutation is about 72% mutation with about 40% mutations in the MHC class 1 gene. So the immune system is totally left in a state where it is unable to really drive an active immune response because the first step of recognition is defective. Now, how are these antigens? Because finally it is the antigen which is going to evoke an immune response. So these antigens have been identified through using cDNA expression libraries or lambda DGT11 libraries. And just one glance at them, you'll see that you've also noticed that these are also present in normal cells. So they're not novel antigens or neoantigens. They're there in the cells involved in normal cellular processes. And they can be classified into 3 regions. And these are the cancer germline antigens expressed in immune-privileged germline cells. These are the melanoma-associated antigens, the NY-ESO, very much expressed in oral tumors, esophageal tumors. Cell differentiation antigens, which are expressed in adult tissues. These are tyrosinases, GP100, MART1, prostate-specific antigens, or antigens which are overexpressed in cancer, HTERT, HER2, in breast cancer, mesothelin, and MUC1. Now, so basically, these are antigens which are self-antigens, but which are either mutated or highly expressed in certain tumors. And as I told you, in the novel area or the new area, which now scientists are pursuing, is the neoantigens. And this has come from the work of the exome sequencing, because tumors are analyzed for non-synonymous somatic mutations. And RNA-seq data, which is available, will focus on mutations, these mutations expressed in specific genes. Peptide stretches containing these non-synonymous mutations are generated by in silico approach. And then you have these computational algorithms which can define peptides which can fit into the MHC, the antigen presenting molecule, which will fit into the groove of the MHC. And these peptides then are the novel antigens which are going to evoke T cell responses. So more the mutation, more the ability to generate new antigens. And now the entire vaccines development is based on fishing out these new antigens, which can, of course, evoke a good T cell response against the tumor. Now, another molecule which is targeted by the tumor is a co-stimulatory molecule, or the co-stimulatory signal. Let's first understand what this co-stimulatory signal is. As I told you, this is a T cell, and this is an antigen-presenting cell. Imagine an antigen-presenting cell is like a tumor cell, because tumor cells are also able to present their antigens. So an antigen, as I said, can be presented in the context of MHC class 1. So this is a processed antigen, which is sitting in the groove of MHC class 1, which is recognized by the T cell through the TCR. It's a heterodimer made up of an alpha and beta chain hooked on to an intracellular signaling molecule, which is the CD3 zeta. If the antigen presenting cell or the tumor cell engages with the tumor cell, via the MHC TCR, of course, with the antigen presented in the group by the antigen presenting cell. This is called as signal 1. And then you need a second signal, which is generated through a molecule, which is called a CD28, which is expressed on the T cell, which engages with CD80 and 86 on the antigen presenting cell or the tumor cell, which is the signal 2. To have an active T cell response, you need both the signals. You cannot have a T cell which gets activated or becomes functional in the absence of a co-stimulatory signal. So this is the basis of a generation of a T cell immune response. And this is what the Nobel Prize, and which I'm going to talk about and why it was given, and I will come through, which I'll be explaining to you in the following slides. So what the tumor does, as an escape mechanism, it lowers the level of co-stimulation. So as I told you, tumors are not professional antigen presenting cells, but they express all these molecules. So tumors, as I said, both the signals are needed. What the tumor does is it downregulates the co-stimulatory signal. So once the co-stimulatory signal is absent, the T cell will not be able to proliferate. It will, in fact, go into a state of apoptosis or energy or get exhausted. So remember, two signals are needed, and the tumor is very well able to offer low levels of co-stimulation. This is exactly what happens. This is a T cell. And as I said, the T cell engages through MHC TCR complex and the co-stimulation. Now, in a T cell, if these two signals are present, the T cells will proliferate, they will produce a lot of cytokines, but you don't want this response to go on unabated, because that would be deleterious. So you need some control, some brake on these cells. And that is mediated through molecules called program death 1 and program death ligand 1. The PD1 is expressed always on an activated lymphocyte, and the PDL is on the antigen presenting cell. So when an event occurs, that is when an antigen presenting cell presents the antigen and T cell gets activated, there's a response, but then it needs a break so that the response doesn't go out of control. So this PD1, PDL1 is hijacked by the tumor cell for its own benefit. So tumor starts expressing PDL1, tons of PDL1, which engages with the PD1, which is present on the immune cell. So imagine, the tumor cell puts a brake to the immune cell, dampens the immune response. And this is antibodies, or the checkpoint inhibitor antibodies do. They block the PDL1 and the PD1. Now, besides the PD1 and PDL1, there are other molecules. There is a list of other molecules, and I'm not going into the details of all this, which also the tumor cell uses. So now there are a lot of antibodies which have come up to block some of these molecules, and which are already in clinics or in clinical trials. So which are these antibodies? We have the CTLF4, which I told you, one of the molecules which engages itself. There's ipilimumab, which is used in the treatment of lung, kidney cancer, prostate cancer, and several cancers. They are in phase 3 trial. They've also now offered, it's already come out of phase 3. It's given as a treatment modality. You have PD1, you have pembrolizumab, which is offered in the clinics. You have nivolumab, which is offered in the clinics. All these are now crossed phase 3 and now offered for the patient benefit, and they've shown remarkable responses in treatment refractory cases, as I mentioned earlier, and long-term durable responses. This is important. These patients have shown long-term durable responses, which was never seen before, not seen with standard modalities of treatment. But again, I wish to emphasize that this is seen as a subset of patients, and we need to understand why some of the other patients are not responding. Again, I come back to the mutational load, and this has been emphasized a lot, because patients who have a higher mutation load show better responses to checkpoint inhibitor antibodies. And this is cases of malignant melanoma, the skin tumors, which were treated with CTLF4. And of course, if there are more mutations, this is a Kaplan-Meier graph, which shows that a survival benefit to patients whose mutation load is high compared to those who show very low mutational burden. And again, I just remember that I told you that mutation load is related to the expression of neoantigens in these tumors. More neoantigens, more is the immune response. And this is again beautifully shown, which by paper. These are all landmark papers, and those who are interested may please read these papers. It was shown that they took a battery of tumors and looked at the mutations, and they said these tumors, the colorectal, skin, lung, bladder, esophagus, stomach, head and neck, which show high mutation load, also responded well to the checkpoint inhibitor antibody therapy, nivolumab, atezolizumab, or pembrolizumab, compared to those which had a low mutational burden. So definitely, the mutational load in the patient also dictates its response to checkpoint inhibitor antibodies. And this again, this is showing that these patients with high frequency of mutation or a high mutation load also show a higher repertoire of neoantigens. So it all goes well in hand, that high mutation load, high expression of neoantigens, and better would be the immune response. And when you say immune response, naturally, it would be the checkpoint molecules which will be expressed, and naturally the response to checkpoint inhibitor antibodies. Now, besides these, the tumor also recruits, as I said, a lot of immune suppressive cells to the microenvironment. And I'm not able to cover all the cell types, but I've taken up key examples. One is the immature myeloid cells. You have the tumor-associated macrophages and the regulatory T cells, which, as I said, through the chemokine and the cytokine signals, the tumor invites them into the tumor microenvironment. Now let's look at myeloid-derived suppressor cells. What are these myeloid-derived suppressor cells, which are called as MDSCs? And a lot of attention now on these MDSCs, how to stop the activity of these MDSCs, how to stop their recruitment to have an active response to active outcome of immunotherapy. In normal hematopoiesis, the hematopoietic stem cells get differentiated to common myeloid progenitor cells and then to immature myeloid cells, and this happens or gets regulated through a battery of cytokines, which is GM-CSF, M-CSF, SCF, IL-3, and FLT3. These immature myeloid cells go and harbor in different tissues of the body, where they mature as macrophages, dendritic cells, or as granulocytes. Now, what happens in cases of certain infections and traumas, these immature cells accumulate, and they do not differentiate, but they accumulate as immature myeloid cells, and they have the property of immunosuppression. They differentiate into what are called as tumor-associated macrophages, the bad M2 type macrophages, which also contribute to immune suppression. So remember, MDSCs are basically immature cells which are found during the course of infection, trauma, and also accumulate during tumors. Now, for a long time, it was very difficult to identify what these MDSCs are, because do they have specific signatures? And we immunologists are very fond of what are called as CD molecules, the cluster differentiation antigens, to identify, because there are antibodies available, and you can do multicolor flow cytometry to identify these cells. So a lot of work went and a lot of discussions went on whether what are myeloid-derived, whether they really exist, and what type of myeloid-derived suppressor cells are present. And finally, we have the signatures. We know you can use these markers. For example, talk about the human phenotype. They are CD11b positive, CD14 positive, CD15 negative, and these are the MMDSCs. That means they're the monocytic MDSCs. We have polymorphonuclear MDSCs, which are CD11b positive, CD14 negative, which is a marker for monocytes, and CD15 positive, and both are HLA-DR negative. Once we have these in hand, once we know how to identify them, we can screen several tumor types to identify where they are present and what level these MDSCs are present. And once you know they are present, you'll know also how to get rid of these MDSCs. And very interesting data that MDSCs also act as independent prognostic markers. If you look at cancer patients, even before you start treatment, if you look at the level, and this was a study done to show that these MDSCs, say pancreatic cancer, esophageal cancer, gastric cancer, and they looked at survival of MDSCs, and those which had MDSCs which are more than 2%, and those which are less than 2%. Those which are more than 2% did not do well. The survival was not good, while with the less MDSCs, the survival was better. Therefore, unit increase in MDSC percentage was associated with that increased risk of death. So you can imagine how important this cell type is. Now I'll share with you some of the data from our own lab where we are profiling patients of oral cancer. Because we have been studying, my lab has been studying for the past several years the role of an immune cell called as the gamma delta T cells. And we want to understand how, because basically it's a killer cell. It has all the machinery to kill the tumor cell. So we are interested in taking this ahead. And if we are to understand the role of gamma delta T cells, we have to also understand the tumor microenvironment and to also understand what immune suppressive cell types which can impede the functions of gamma delta T cells are present. So naturally we profiled and we showed, I'm not going into the details of the markers, but just I'll tell you that they are present in the oral tumors. They also infiltrate in the tumors. So this is, when you get a surgical biopsy, we are able to make single cell suspensions. We are able to do extensive flow cytometry to identify the MDSCs. This is published, and those who are interested can read this paper. And we see that it is stage related. From stage 1 and 2 to stage 3 and 4, the levels of MDSCs increase. Then the classical experiments of co-culturing the T cells with MDSCs. And as you add MDSCs, the T cell proliferative response goes down. So MDSCs have the ability to inhibit proliferation of T cells. They also have the ability to inhibit secretion of interferon gamma. So this is a T cell without MDSC, which would an activated cell which would secrete interferon gamma, which is a key cytokine in the entire cascade of an immune response. And you add MDSCs, and this MDSCs dampen the interferon gamma production. What these MDSCs even do is they allow and promote the differentiation of another cell type, which is called as Th17. And this Th17 contributes to inflammation. And we have gone more in depth to show that MDSCs induce Th17 and use the nitric oxide synthase and the COX-2 pathway, clearly showing that Th17 production is reduced if you add LNMA, which is an inhibitor of NOS, or you add NS398, which is a COX-2 inhibitor. You see that there's so I'm not going, as I said, I'm not going to details of all the experiments, but the take-home message is that MDSCs can induce Th17, another cell type, which can also be, which can further add to tumor progression. And here again, I take the example from one of the papers which we published in gallbladder cancer patients, where we showed these were delta 17s, that is the gamma deltas, which were Th17 producing cells. They inhibit, when we did these experiments, we found that in fact these cells promote VEGF production, the vascular endothelial growth factor production, which promotes angiogenesis, which promotes the blood supply to the tumor cells. And this is the entire work doing angiogenesis array to show that if you take the IL-17, which is secreted by the delta 17 immune cells, these can promote angiogenesis. We've done a lot of biological assays, also the CAM assays, to show that it promotes angiogenesis. It's also related to survival because patients who showed, because these are gallbladder cancer patients, these are patients who showed high levels of gamma delta 17 did not do well compared to patients who had low levels of gamma delta 17. And of course, these are others with the interferon gamma, you just see the reverse. So interferon gamma is like the good cytokine, and the IL-17 producers are the bad ones. And we were able to do this study in the gallbladder cancer patients. So in short, MDSCs are very dominant immune suppressive cells, but their recruitment, their accumulation, their trafficking can all be suppressed using specific molecules which are inhibitors, like thrombin inhibitors, COX-2 inhibitors, and a whole lot of them which have been identified and are in clinical trials now to block MDSCs. So MDSCs can be blocked. In fact, people are talking of combination therapies. While using immune checkpoint inhibitors, or if you're using CAR T cell, you also block the MDSCs to get an active immune response. Now, coming to the other cell type, which I which I want to talk about are the monocytes. And in the monocytes, you have two types. Again, you are the good ones, the M1 macrophages, which have bactericidal activity, inflammatory cytokines, immunostimulatory, which lead to tumor suppression, and the M2 macrophages, which in fact promote tumor progression. And one needs to identify the M1 and the M2 type. So it is important to block these TAMs, or the tumor-associated macrophages, in the tumor microenvironment. And how the TAMs mediate immunosuppression? They light up the PDL1, which I explained to you, which the tumor also hijacks. They light up the PDL1, VISTA, B family molecules, and because of this, they can dampen the CD8 responses, which is the killer cells, the CD4 responses. They can invite the regulatory T cells, because they secrete a lot of interleukin 10 and TGF-beta. And the regulatory T cells, which are immune suppressive cells, they invite them into the immune microenvironment. They dampen the DC responses, and they call a very interesting phenomena, which they called as the metabolic starvation, which is called as metabolic starvation, by secreting arginase and indolamine dioxygenase. And this is actually a tryptophan quencher. Tryptophan is needed, amino acid needed by the T cells for its growth, for its functionality. And these TAMs can secrete arginase and idose and can inhibit T cell function. And of course, can also lead to induction of fibrosis by activating the CAFs, the cancer-associated fibroblasts. Now, I'm not going to the details of it, but you can stop the recruitment, immunosuppressive functions, and polarization of these TAMs by using there are a battery of cytokines and chemokines which have been identified. Another regulatory T cells which I want to talk about are the regulatory T cells. These are our dominant immunosuppressive cell types which have a specific signatures. These cells have express CD4, CD25. These cells, it's not working. These cells express CD4, CD25, which is an IL-2 marker, and CD127 low, express the transcription factor FOXP3. They represent they're 5 to 10% in the periphery. They express all the PD1, the CTLA4, the GITR, TIM3, all the markers which I mentioned to you earlier, which can engage themselves and dampen immune responses. They secrete tons of IL-10 and TGF-beta, IDO and adenosine, again, all inhibitory molecules. And they show other expression of other TNF receptor superfamily markers. So to elicit a good anti-tumor immune response, you have to take care of these regulatory T cells. And of course, there are agents which can block these Tregs. There's one agent which is called as denileukin difetitox, which is the blocker of the CD25, which is expressed on the regulatory T cells, or cyclophosphamide also is used to block the Tregs. Now, we did this study to look at our patients, Indian patients, and we showed that they have the tumor infiltrating lymphocytes they have a high percentage of regulatory T cells which are already present there. And again, here it is stage dependent. They're present at different sites in the buccal mucosa, in the tongue, and any other sites. So they are all over as immune suppressive cell types which are present in the oral cancer patients. So imagine it's challenging for an immunotherapy, because already these immunosuppressive molecules and the cell types are present in the tumor microenvironment. This is again the data from our lab, where we are looking at cervical cancer patients, and we stage them according to the FIGO classification. And as the stage advances, you see a marked increase in the regulatory T cells. I'm not showing you the data, but we have studied in detail how these regulatory T cells interact with the immune cell types, both in the oral tumor microenvironment and in cervical cancers, and how they cross-react with the immune cell types. Again, there's enough data to show that these T regulatory cells, they can predict prognosis, because if you're high number of these cells, say it in hepatocellular carcinomas, and this is a case of ovarian cancer, if they're high in number, there's poor survival. And if they are low in number, the survival upfront, you can know that the survival of the patient is better. Very quickly, I'll tell you how these regulatory T cells work. As I said, they secrete TGF-beta, which inhibit effector T cells, they secrete molecules such as granzyme A and perforin, which can literally drill holes into the membranes of the T cells, and the osmotic balance can be lost. They are quenchers of IL-2 because they express CD25, and any activated immune cells will secrete IL-2 and would require IL-2, which is quenched out by CD25. They also release a lot of immunosuppressive molecules like adenosine, and they engage and block through the co-stimulatory molecules. They block the maturation of the DCs, or the DCs secrete endolamine dioxygenase. So these are the various mechanisms which the immune suppressive cell types use. So in the end, I want to just say here that this is an immunosuppressive network which is created in the tumor microenvironment, and there's a constant crosstalk going on between these cells. MDSCs has a central role in this, and these, as I'm not repeating all things which I've said earlier, but MDSCs play a key role in differentiating into help in differentiation of Th17 cells, which are pro-angiogenic and help and promote tumor cell growth. They also dampen through arginase, iodos, and all the cytokines through reactive oxygen species, and bring down the loss of T cell signaling in key adhesion molecules, interferon gamma, caspase, perforin, granzyme B, and we have shown this experimentally, and we have almost dissected the immune suppressive microenvironment. So this is the challenge now for a classical immunotherapy to work. It has to cross all these barriers. And now the second half, which I'm going to talk to you about, is about immunotherapy. So in the first half, we try to understand how the challenges thrown by the tumor cell, and now we'll try and understand how we can exploit the immune system to fight against these tumors, which are, as I said, very smart. Now, this was William Coley, father of cancer immunotherapy, and way back in 1800, William Coley, actually, he was a surgeon, and he was treating patients with osteosarcoma, bone tumors, and he injected into them pus cells, and he saw wonderful regressions. Nobody, of course, believed it, and he faced a lot of criticism those days. But these were identified as bacteria, which are Serratia marcescens and Streptococcus pyogenes. And after years of neglect, scientists are now seeking to replicate his success. And this has taken a lot of interest now in using these approaches for treating cancer. So there are different modalities for treating cancer. There are non-specific immunotherapies which we can use, like BCG, levamisole, or monoclonal antibodies. You can have vaccines, and of course, cell-based therapies, which I'll be describing to you soon. Monoclonal antibodies, most of you must be knowing, this George Kohler and Caesar Milstein, who discovered these antibodies, and I was at the Max Planck Institute when he was awarded the Nobel Prize. And what they discovered was they were interested in somatic hybridization, or making somatic hybrids, and they discovered the way to make mouse monoclonal antibodies. Now, there was a lot of euphoria generated when mouse monoclonal antibodies, which are directed against tumor antigens, were made, because people thought now this would be used to treat cancers. But unfortunately, there were a lot of adverse reactions because of the HAMA response, the human anti-murine antibody response, which was generated against the mouse proteins. So it was important to engineer these antibodies to make these antibodies more and more human. So what they did was have the variable regions from the mouse on the backbone of human FC regions, and then they further engineered it just to have CDR3 regions, the complementarity determining regions from the mouse and rest of the backbone of human. And finally, they're able to make the fully humanized antibodies. So these antibodies are in the clinics. There's a list of antibodies which are being used in the clinics. And some of you may be familiar with the names like cetuximab, bevacizumab, or Herceptin, which is regularly given now to treat breast cancer patients. Rituximab, which is directed to treat patients with leukemias and lymphomas, which recognizes CD20 antigen, which is present on the blasts. Monoclonal antibodies can be tagged with cytokines, they can be tagged with radionucleotides, with toxins, and then can be administered. There's a beautiful way in which you can use SCFP, the single chain antibodies. You can tag them with radionucleotides, the indium-111 or technetium-99. And there's a technique called immunoscintigraphy, which we can use to look at tumors, which will be called as occult tumors, can be detected using immunoscintigraphy. So a lot of applications of the monoclonal antibodies. Now we come to the CAR therapy, and this is where a lot of interest now is there. This is the chimeric antigen receptor T cells. This is a chimera created on a T cell. Chimera because it uses the property of the BCR, the B cell receptor, to recognize a wide array of a repertoire of antigens. They use that receptor and hook it on through a hinge region. And this is a transmembrane region of the T cells, and hook it on to the endodomains, which are made up of these molecules, which I explained to you earlier, the co-stimulatory molecules, and then to a signaling molecule in the T chains of the T cell. So basically, it's a chimera. The antigen recognition property from the B cell hooked on to the signaling in a T cell. And this is called as the CAR T cell. There are already two which are FDA approved, which have come into the market, and this is Yescarta and Kymriah, which is used for the treatment of relapsed or refractory large B-cell lymphomas. They've shown amazing results in patients, and also used in acute lymphoblastic leukemia. So both these two are now already available. Of course, it's a prohibitive cost. If I tell you the cost, it's $400,000 to treat, which is absolutely unaffordable. But there are efforts now, even in India, to prepare indigenous CAR T cells. And these CAR T cells, there are several generations, there are several modifications have been done, and these are in the intracellular domains of the CAR T cells to improve its antigen recognition ability and its signaling in cells. We now come to the 5th generation of CAR T cell, where you can block the endogenous alpha-beta receptor, which is present on the T cell, giving an advantage only through the engineered CAR T cell to recognize the tumor antigen. This is the way how you manufacture CAR T cells. This is very simple. By the process of leukapheresis, you take the lymphocytes out. You have the ways of separating, of activating, stimulating, expanding these T cells. And there is a process of transduction where you introduce the CAR construct. These can be viruses, lentiviruses can be used, or other retroviruses can be used. And then finally, you harvest the product, which comes out, which is directed against a specific tumor antigen. Now, there are a lot of interest now on what to target. See, the CAR T cell has shown interesting, amazing results in hematological malignancies. But the challenge is the solid tumors, because to cross the solid tumor, to enter into these tumors itself is a challenge. So a lot of work now is going on these CAR T cells, targeting these various antigens, the EGFR, GPC3 in hepatocellular carcinomas, the MUC1, HER2. So both for leukemias, I said it's a success story, or it has shown, it has worked very nicely. Solid tumors, still a question mark, and a lot of work going on in the solid tumors. I'll skip this one, and we come to the vaccines, which is the final part of this talk. Vaccines, everybody is interested in knowing, are there vaccines for cancer? Now, for other infectious diseases, you have prophylactic vaccines. You take them, when you are normal, you take them so that you are protected because of the long-term memory which is generated, or the neutralization. But in cancer, there are no prophylactic vaccines. The only example of the prophylactic vaccine where it's against the virus, human papilloma virus, which is associated with cervical cancer. But all the other vaccines are therapeutic vaccines. So the challenge for the vaccine here is already the tumor load, or the tumor is existing, and it has to now boost the immune response against that existing tumor. A lot of attempts have gone in making tumor cell vaccines using autologous or allogenic tumor cells, using dendritic cell vaccines, peptide vaccine, antigen vaccines, anti-idiotypic vaccines. Each one would form a lecture on its own: DNA vaccines, mRNA vaccines, and viral vaccines. What has really reached the clinic is the dendritic cell vaccines and the first cell-based vaccines. I'll talk of the OncoVax, which have worked against the colon cancers, along with admixed with BCG in stage 2 colon cancer, approved in 2006 by the FDA. OncoVax reduced by 44% the risk of patients, cancer recurring in these patients, and it benefited stage 2 patients. But still today, we do not have enough data to show that it will work in all types of colon cancers. Researchers are still phase 3 trials, 3B trials is going on, and they're recruiting patients, and we need more data on this vaccine. Again, the cost of this vaccine is $93,000 for the entire course of the vaccine. Then comes the dendritic cell. This cell is an antigen presenting cell. It's a beautiful cell because it has a lot of properties that it expresses tons of MHC molecules. It expresses co-stimulatory molecules. So it can activate CD4, CD8 T cells very easily. It can secrete the favorable cytokines like interleukin-2 and IL-12, and it can be prepared very easily. Ex vivo preparation that you do. You can take patients' blood, you can harvest dendritic cells, you can mature them using GM-CSF and IL-4, and then you can throw in whatever you want, whatever antigen you want, peptide, antigen, RNA, DNA, and you can easily make it into a vaccine. So this cartoon, it precisely describes what I've just mentioned, taking the monocytes, peptide loading of the dendritic cells and pushing them back. You can also put in tumor cell lysates into them and directly load them with the tumor cell lysates, and these can be given to the patients. This strategy was used, and there was a prostate vaccine, Cellular, which was, or Provenge, which came for the androgen-independent prostate cancer in 2005. It showed a good response, 52% increase in survival, but now it's again the cost was prohibited. Dendron went bankrupt with this vaccine, and it's, as I said, again an unaffordable vaccine for the layman or the common man. There are oncolytic viruses which are now being used, and these viruses attack the tumor cells. They replicate inside the tumor cells and kill the tumor cells. And and there is another bystander reaction because of the lysis of the tumor cells. The T cells surrounding the tumor can get activated. So oncolytic viruses, a lot of interest now, and these are being pursued. And the viruses which are used are adenoviruses, herpes viruses, and they have gone, if you look at the color code across, you'll understand that they've been used as combination therapies, and they are in clinical trials. And of course, this color is the adenovirus. So a lot of work in phase 1, phase 2, phase 3 clinical trials with the adeno-oncolytic viruses. The future today, if you look at, is definitely going to be personalized vaccines, which would be the expression of neoantigens, which would go through, as I explained to you, the tumor cells, through all exome transcriptome sequences, fish out, use the predictive algorithm, fish out the right peptides which can fit into the MHCs, and use different modalities for vaccine delivery. And here nanotechnology plays a very important role. A lot of trials going on with these nanovaccines, putting them back and getting an active immune response against these tumors. So this is the future, and a lot of work, not a lot of vaccine scientists now getting engaged into this discovering of the neoantigens and generating vaccines. I'll end up with only 2 important points: the importance of the gut microbiome and the response to immune checkpoint inhibitors. This was a very interesting paper which came in Science from Gopal Krishnan et al., who showed that higher gut microbiome diversity was associated with improved response to checkpoint inhibitor antibodies. So your gut microbiome dictates your response to immunotherapy. And they were able to actually, they took patients, melanoma patients who were on the immune checkpoint inhibitor therapy, the PD-1 therapy, and they classified these patients as responders and non-responders, and then looked at their gut microbiome. And they could identify the favorable gut microbiome, which had high diversity, the Ruminococcaceae and the Faecalibacterium, and the non-responders showed low diversity and relative high abundance of bacteroidetes. So you have here now a tool in hand, the gut microbiome, which can dictate your response to checkpoint inhibitor antibodies, opens up in understanding the gut microbiome. In fact, fecal transplants are being pursued along with checkpoint antibody inhibitors and along with immune therapies to see the outcome. And the last one is the metabolic imbalance and response to checkpoint inhibitors. In the tumor microenvironment, you have a tumor cell which has a high demand for glucose, relishes glucose, and this poor immune cell, which has to proliferate, which has to show its functionality, also needs glucose. From oxidative phosphorylation has to shift to glycolysis, but the tumor cell depletes all the glucose stores. In fact, makes it because of hypoxia, HIF1 alpha expression has a lot of inhibitory effect on the cytotoxic T cells. And we need to understand all these metabolic pathways. And there are inhibitors for all this, the glutamine, the glutamate pathway, arginine path inhibitors, IDO inhibitors, and a whole list of them. But these are still in preclinical stage and are being tested. So how can you improve a metabolic imbalance so that the response to immune therapies would be better? To summarize what I've said, the tumor cell has a profound influence on the immune cell by downregulating MHC class 1, loss of antigenicity, loss of co-stimulation, recruiting immune suppressive cell, release of immune suppressive factors, altered metabolism, and also upregulation of STAT3 transcription factor. Now imagine this negative influence. Is there a ray of hope? Of course there is. And you have different modalities which have come up, the checkpoint inhibitors, the CAR T cells. Now, not only CAR T cells, you have the chimeric engineered T cells, they are engineered NK cells, they are engineered NK T cells, they are engineered gamma delta T cells. There are adoptive cell therapies which are unmodified T cells like NK cells and gamma delta T cells. Combination therapies used in combination with radiotherapy and radiotherapy and chemotherapy. There are vaccines, which I explained to you a few minutes ago. And then, of course, understanding the gut microbiome and the metabolism. So we have these tools in hand, and I'm sure there's a long way to go, but still there is hope. And immunotherapy has certainly paved out a new pathway for taking care or for eliminating the tumor cells. Thank you.
So that we can take questions.
Yeah.
Thank you, Dr. Chiplunkar, for handling a very, very complex subject in a manner that you can explain the complete range in 1 hour. I'm sure there will be a number of questions from the participants. So we have requested the participants to post the questions on the chat mode. And we will wait for the questions. Maybe to start the discussion, being a complete non-specialist, I can ask some questions which may be bordering on the silly but related to this topic. In the context of this COVID infection, everywhere people have been advising, first thing you have to do is improve your immunity. So in the context of your talk, what exactly happens when you say that I have improved my immunity? I eat this, that, etc. I have improved my immunity. In the body, what does exactly happen which can fight a new organism?
A very nice, very interesting question, which is in everybody's mind. Because improve your immunity, it's such a broad term which has been used, and people promote so many agents which can improve immunity. Do we have a handle? Do we handle a measure to say that this is good immunity? See, there are immune cells. We don't have a single test which can tell you. There are broad, you can look at the lymphocyte counts, you can look at CD4, CD8 ratios, but that cannot predict and tell you upfront that your immunity is good or your immunity is low. Unless you have a specific antigen against which you are testing. You're looking at the COVID antigen, and then you are measuring the immune response. You can define immune response. But in general, it's very difficult. There is no measure which can say you have a good level of immunity or you have a low or bad level of immunity. I'll take the example of HIV, where CD4 cells get depleted and patients become immunosuppressed. So there are counts there which you can say below certain level, below certain threshold, if your CD4s are there, you're immunosuppressed. But otherwise, in a healthy individual, to define that or say that you are a person with good immunity and you're a person with low immunity, it's very difficult to define.
But my question is slightly different. How is a person who is more immune compared to another person? How does in his cell system, what is the difference between, that is what I want to know. What exactly happens when you say I've improved my immunity? What exactly would have happened?
It is basically your first, the infection is taken care by your antigen presenting cells. They've handled your antigen very nicely and activated your T cells. So your T cells have been able to take care and eliminate this infection. That is when you say, and not only T cells, also the cells of the innate immune system, because I told you there are two arms, the innate and adaptive. So these have acted to their efficiency to eliminate the infection.
Okay. So now you can see there's a question on some chemotherapeutic medicine.
Rituximab have also been used for other conditions. Yeah. Can it be other way, or are there any studies in this direction? Medicine like rituximab, wherever rituximab is directed against CD20. So if there is CD20 expression, rituximab can be used, and this can be used in combination. There are antibody-dependent cellular cytotoxicity. You can hook on to an immune cell to target the antigen where it is present.
Can you change it to Defects? Just one, let's we will help you. Yes, there are some questions here. Yeah. I understand that the response on the patients varies widely for each type of treatment. Could there be other factors such as the mental attitude, stress, etc., which are non-measurable that influence the treatment outcome? How does one address this, and is it possible to increase the immune response?
Of course, there are other factors, and as you rightly mentioned, stress, and there are ways and measures, not directly, but you can measure that the stress does influence the immune response. The catecholamines, there are so many other things which are released during stress, endorphins which are released. We can measure these, and you know that stress has a profound influence on the immune system.
Any other questions from the audience?
Yeah, I have a question.
How much scope you see in a universal antigen based here? Universal antigen based vaccines and CAR therapy. Do you think that the personalized vaccines and CAR therapy are future perspectives?
There is a whole lot of interest in CAR T cells. As I said, identifying the right antigen is important. Now, if we are able to identify driver mutations and you're able to predict the epitopes, which are because, or predict the peptides, which are because of these mutations, there could be a common vaccine, but otherwise it would be like a personalized vaccine for each patient. And we need to have a universal vaccine. That would be the right way to go. There is a new paper which I just read, which is on cytidine deaminase, which is being APOBAC, which they say, introduce cytidine deaminase into tumors, which will cause a lot of mutations and make the tumor immunogenic. And this is one of the newer approaches which is going to come up. CAR T cells, as I said, unaffordable treatment, but since it has shown remarkable responses in leukemias and a lot of, in India itself, there are scientists and institutes which are working towards it. I feel an affordable CAR T product would be soon coming out. Can you talk about microenvironment of normal versus cancer cells? How it is related to human gut bacteria, and is there any relation with drug resistance? Yeah, the microenvironment of normal versus cancer cell. In normal, there is, you see, you cannot compare what is a normal microenvironment. Normal microenvironment are normal cells which are present. Tumor has a microenvironment because these are stromal cells, and there are other cell types, the immune cells, and I explained to you all the immune cells which are present, which are in close proximity of each other. So definitely the tumor microenvironment is very, very different. And I don't understand what you mean by a normal microenvironment, but tumor microenvironment is very different from, say, even if you compare it with the peripheral blood, it is very different. How is it related to human gut bacteria, and is there any relation with drug resistance? Drug resistance and gut bacteria, I don't have the answer to this because there is not much work which has still gone in to understand drug resistance and gut bacteria.
Okay, I want to ask a provocative question again as a non-specialist. I was seeing a discussion on one of the TV channels regarding the vaccines being developed for this COVID. And one of the speakers mentioned that India is excellent in making vaccines, and world over we are supplying vaccines, so that's something to be proud of. But we are making vaccines based on somebody else's prescription. Is it correct? And what they said is that what we need to do in India is to develop the methodology, technology, expertise, and required infrastructure to be able to make vaccines. Subsequently, producing them in large quantities, we are already doing very well. But why is it that we are not able to take leadership there?
See, as you rightly said, using somebody else's um, uh, idea and making the vaccine. There are two components to this. I don't know whether I should take names, but Serum Institute of India is the largest producer of vaccines supplying in the world over. But again, it is importing the idea from somewhere else. But there's another company which has come up, which is going to make a thermostable mRNA vaccine, and it is indigenous. And again, it's from Pune. So the effort is on, and I'm sure there will be an indigenous vaccine which is coming up.
Well, I will wait for, there are some more questions coming up. So one more question from a non-specialist is, we are reading all the time about this vaccine has crossed stage 1 trial, stage 2 trial, stage 3 trial, etc. So what is, can you just explain to a non-specialist, what exactly is that stage 1, 2, 3 trials? I mean, is that stage 3 trial is required for every vaccine to come to market? What is it?
Because see, when the vaccine is to be administered to the general public and it has to go through several levels of testing. In the phase 1 trial, it is a smaller number of patients, the safety and efficacy testing of the vaccine, a smaller number of patients. When it has crossed that, it can go to phase 2, where the sample size then increases. And then in stage 3, it is still much, much larger sample size. So in doing that, you come to know the adverse reactions, the serious adverse reactions, which are, because it involves follow-up, a long follow-up. So serious adverse events, serious adverse reactions get noted in that. You cannot be on a fast track for making vaccines. Still today, it's surprising to see we don't have vaccine for malaria, we don't have vaccine for dengue, we don't have vaccines for JE, which are, vexing problems in India. So unless the vaccine has done all these phase 1, 2, and 3 testing as per the guidelines, it is difficult to accept it as a safe vaccine.
So there's a
Hello.
question with respect to connection between
So it's
blood glucose
this
and cancer.
Yeah. What is the question? Connection between blood glucose. Metformin is one of the which is given to diabetic patients. Is also known, a very interesting. This is also being used as one of the metabolic inhibitor because to inhibit the glucose.
So, person wants to know, often certain cases are known only when it's a third stage. Is there any way to?
At early stage. This is a very nice and an interesting questions. Not all cancers can be picked up at early stage, because I'm not talking only of early diagnosis or prevention or going and checking yourself regularly. That's a different thing. But some of the cancers, there is a pre-cancerous stage associated with these cancers. Take the example of oral cancer, where there's a lot oral cancer arise because of tobacco abuse. Now, these early lesions, precancerous lesions called as leukoplakia, erythroplakia, these can easily be detected. The white patches which come in the mouth can easily be detected. They can easily be excised. So there is a precancerous stage which is there. In cervical cancer, you have the SIN lesions, the precancerous lesions. You go for a checkup and it will be in the early stages. So these SIN lesions can be identified and a treatment can start earlier. So they're not there for all the cancers known, but yes, there are precancerous stages for some cancers and which you can treat.
What yes. Yes.
The beginning you were showing some symptoms, kind of from there you have to be careful.
Which I think
Symptoms of cancer.
Symptoms. Huh?
Yeah.
Huh.
So, different reasons. How one can say that I have all kinds of problems.
No. See, the simplest answer is, if you say hoarseness, I gave the example of hoarseness, or prolonged cough or something. See, if it is treated, you don't have to immediately when you get hoarseness doesn't mean it's there. But if it's a prolonged duration, it doesn't, with standard medication doesn't go away.
What?
Then you have to alert yourself. Or there is a wound which does not heal. With normal medication, normal treatment doesn't heal. It is the time, you should, or a lump which you notice. There, I would say you don't even wait. You want to. In the breast for females, if you self-examination, you notice that there is a lump which you can feel, palpate. It is better to then immediately go.
Okay, there's a question which is a technical version of my question. I would not have, I asked in a different manner. Somebody asked more clearly. When we say there are natural ingredients that boost our immunity, in which cells exactly are they enhanced? Immunity is enhanced. Does the response of T cells or APC, which gets altered?
No, see, these are called as immunomodulators. And these immunomodulators may have different effects. Some immunomodulators affect both innate, which I explained to you, and adaptive immune responses. Some only specifically work on T cells, and that too on a subset of T cells. So unless all this is studied carefully in a lab, like I'll give you example from my own work. Now, we all consume black tea and green tea. There is ethylamine, theanine, ethylamine, which is present in the black tea. Now, these things are able to stimulate or activate gamma delta T cells only, not other cell types. So gamma delta T cells get stimulated and innate immune cells by the components which are present in, and we did a collaborative project with Unilever just to understand this more in detail. So yes, immunomodulators cannot, it's not just, working on all immune cell types, they can also work on specific cell types. But you have to do this work in the lab to understand which cell types are getting activated.
Okay, there's a question on if we make tumor cells
highly immunogenic
to get targeted by immune system, won't it have an adverse effect on the body in general?
Yes, I gave you the example of cytidine deaminase EpoBac, which is now the latest paper, which is talking of this modality. There is, like you have, it is site-directed or cell-directed. There are ways now you can do it very specific. There are intratumoral injections which you can give. So it only goes to the tumor cells. And so you can have, it might disseminate to some extent, but a depot will be created in the tumor cells. You can do this. There are so many ways in which you can make it cell specific, and this can be achieved.
I think we will stop here. There will be a lot of questions, obviously, because the topic is of great interest to a lot of people, and also your talk has naturally increased the curiosity of many people. I'm sure that you will be happy to answer any questions subsequently in case they contact you by mail.
Sure.
And
I've put my mail Gmail ID on the first slide itself. I would be happy to answer any of the questions or discuss with you, or if there are any good ideas you wish to discuss also, I would be more than happy to discuss them.
So I think we will come to the end of today's webinar and convey our sincere thanks to Dr. Chiplunkar for very, very deep discussion on the clash between the two titans. Obviously, it has increased the curiosity of many people, and I also would like to meet you and learn from you more about this in future. Thank you very much for joining us and spending your time. And we are also happy that today it went without any technical glitch, because last time you gave the same lecture, halfway we had to stop because of some network issue. I'm glad that this time this didn't happen. And thank you very much once again. And thanks to all the participants for joining us today. Thank you.
Thank you, Dr. Rao, Professor Rao. I really enjoyed giving this talk. I was a little worried that I had such a big topic I had to accommodate in a limited time frame, but I hope I've done some justice to give you a bird's eye view of what actually tumor immunotherapy or tumor immunology is. Thank you.